My name is Taylor Zedeca. I'm a junior this year and I'm majoring in neuroscience.
My name is Zainab Saeed. I am a sophomore this year and I'm a biology major.
My name is Kayla Thowen, and I'm a senior psychology and neuroscience major. We are all really excited to talk about the research that we've been performing in Dr. Askvid's lab this semester. Our lab has been investigating the absence of axonal sprouting that can be seen following lesions in 125-day-old rats compared to the sprouting responses that have been shown to occur in younger 35-day-old rats. Previously, an injury model was used where axons going from the supraoptic nucleus of the hypothalamus to the pituitary gland are cut on one side of the brain. In the 35-day-old rats, axonal sprouting then occurs on the opposite uninjured side of the brain. In the 125-day-old rats, however, this axonal growth has not been shown. So in other words, the brain is able to recover when this injury occurs in 35-day-old rats, but in 125-day-old rats, the brain no longer shows this ability. The broader scale question we are aiming to address is this: what changes in the brain to prevent recovery of neurons as the brain ages? One factor that our research surrounds involves the NOGO protein family. NOGO proteins are neurite outgrowth inhibitors that have been shown to inhibit axonal growth, and this can prevent recovery of neurons. There are multiple isoforms of NOGO. There's NOGO A, NOGO B, and NOGO C. NOGO A is our focus of research because it's the primary isoform that's in the brain. It's found in the myelin sheath that covers neurons, and a receptor called NOGO R has been identified as a binding site for NOGO A.
Since NOGO A has been shown to inhibit recovery, and because we previously demonstrated that there's a lack of recovery in the 125-day-old rat, we hypothesized that there would be an increase in the NOGO A protein in the 125-day-old rat. So first we compared the protein concentration of NOGO in the 35-day-old rat and the 125-day-old rat. A Western blot was performed and the protein levels were standardized to beta-actin. A higher concentration of NOGO was found in the 35-day-old rat brain than in the 125-day-old rat brain. This does not match our initial hypothesis, and at this time, we're unsure of the significance of this decrease. Next, we performed a dual fluorescence in the 35-day-old rats to determine cellular localization of NOGO A. In figure A, NOGO A is labeled in green, and in figure B, the neurons are labeled in red. In figure C, the co-localization can be seen and a yellow color is present. This means that NOGO A is co-localized with the neuron staining. In figure D, we again stained for NOGO A in red and stained for the astrocytes in green in figure E. The co-localization in figure F does not have as much yellow staining. We quantified these results and they are displayed in the graph at G. We found that there was less co-localization of NOGO A in the astrocytes, which matches the visual difference we see between figures C and F.
Next, we looked at an antibody that stained for both NOGO A and NOGO B. We saw similar results in that the NOGO AB was significantly co-localized with neurons. We will see NOGO AB in astrocytes, but there was less of it. However, if you look at the green staining in E, you'll see this circular staining pattern. This is NOGO B specifically staining a blood vessel. NOGO B is the only NOGO isoform found in the lining of blood vessels. We also stained for the NOGO receptor and found pretty much the same thing, where it is co-localizing more with the neurons than with astrocytes. So what does all this mean? As Zay mentioned when she was going through the protein data, we expected to see an increase and saw a decrease, and we still don't know what this means. It may have no connection to the recovery response. We also have another mystery. Our neurons do not have myelin around them, but NOGO, which is found in myelin, is co-localizing with our neurons. We hypothesize that NOGO is actually on the other axons that are connecting with our neurons. And because those arriving axons completely cover our neurons, it makes it appear that the staining is in our neurons. We do want to test this hypothesis in the future, so we will perform additional dual fluorescent experiments using antibodies that stain the arriving axons. Additionally, we need to perform Western blot experiments to look at protein levels of NOGO AB and NOGO R. Lastly, we may perform co-immunoprecipitation experiments to determine if NOGO A and NOGO R are binding to each other.